Okay, here is DNA video part 2, and we're going to start this one by talking about RNA. If you take a look at this RNA molecule, hopefully you can see right off the bat that it's different from DNA in the fact that it's only single-stranded. There's only one side to an RNA molecule, and instead of deoxyribose sugar in the backbone, you have ribose sugar. So you have ribonucleic acid instead of deoxyribonucleic acid. And another one of the major differences, hopefully you can see, instead of AT G and C, you have AU G and C. In an RNA molecule, there is no thymine as one of the nitrogenous bases. Instead, it's replaced with something called uracil. It's another nitrogenous base, but again, RNA does not have thymine, it has uracil instead. Now, adenine is going to pair with U in RNA. So that's something that maybe takes a second to kind of think about. Instead of A and T in RNA, A pairs with U. And then guanine still pairs with cytosine. Everything else is the same except there's no thymine, so adenine is going to have to pair with uracil. You can think of RNA as sort of the messenger. It's single-stranded, it has a ribose sugar instead of a deoxyribose sugar in its backbone, and it contains no thymine, uracil instead. So instead of A and T, A is going to pair with U. If you have this DNA strand, ATAGCG, we can write in the RNA strand as A is going to pair with U. Now, this part right here might get a little confusing because you might say, well, you just said that there's no T in RNA, but in the original DNA strand, obviously there still could be a thymine. In this case, if you have a thymine, the RNA complement will still be adenine because RNA still has adenine, it just doesn't have thymine, so that's why we replaced it with the U here. Now again, we have adenine in the DNA strand. In the complementary RNA strand, we're going to need a U, and then G's and C's still stay the same. So guanine is going to pair with cytosine, cytosine is going to pair with guanine, and again, guanine is going to pair with cytosine. I'm going to backspace once here. Now this is what the complementary RNA strand is going to look like, and it's very similar to how we would do it with DNA. A's and T's are going to pair together, except again, there's no T in RNA, and it's going to be replaced with uracil. And here's an image that summarizes those differences between DNA and RNA. RNA single-stranded, DNA double-stranded. Even though that D doesn't stand for double-stranded, it stands for deoxyribose. You can think of it D, DNA, D, double-stranded. RNA single-stranded, like I said, uracil, DNA has thymine. Adenine is going to pair with thymine in DNA. Guanine is going to pair with cytosine in DNA. In RNA, guanine and cytosine stay the same, and you're going to have adenine pair with uracil instead of thymine. And since a purine always has to pair with a pyrimidine, we know that adenine's a purine. So since we introduced RNA, we talked about uracil, so uracil has to be a pyrimidine. And there are three important types of RNA molecules when we're talking about protein synthesis. The mRNA, or the messenger RNA, that's this one right here, it's going to carry codes from the DNA. It's going to use the DNA as a template. And if the DNA code says ATGC, we know the RNA would say UACG, right? Remember, it's the complementary base pair. The messenger RNA is going to take that DNA template, it's going to make the complementary base pair, and then it's going to travel to the ribosomes in order for the ribosomes to synthesize that protein depending on that DNA code. And then we have the ribosomal RNA, or the rRNA. It's going to help make up the ribosomes. And remember, the ribosomes are like the factory workers of the cell, and they're going to put these different codes together in order to create proteins. And then we have the transfer RNA, or the tRNA. The transfer RNA is going to link the mRNA to amino acids. And remember, multiple, and the amino acid is shown right here, that blue little dot right there. Multiple amino acids linked together is going to help create proteins. We'll talk about this process a little bit more, but I just wanted to introduce those terms first. Now, big picture here, think of the DNA like letters. Letters are going to form words, and words are going to form sentences. That's kind of like the premise of how this protein synthesis works. We're going to take the DNA code, and then we're going to use that to code for amino acids, and those amino acids are going to code for proteins. Now, whatever combination of A's, T's, G's, and C's you have in your DNA, that's going to determine what trait you might have. For example, CAT, if that codes for a protein that makes some sort of organism like this alien monster thing here have fangs, then that determines if it does or does not have fangs. So if it has a different DNA code, it won't have fangs. And it works similarly for us. If we have a certain trait, we can look at our DNA code, and if we have a certain combination of nucleotides, A's, T's, G's, and C's, then it will determine what trait we have. And that represents the process of transcription. In transcription, we have both sides of the DNA molecule here. Then remember, we have that RNA, so the messenger RNA is going to use the DNA strand, one of the DNA strands as a template, and it's going to create an RNA complement to that. Then that mRNA is going to go off to the ribosome. So when you're transcribing something, think about like a court reporter. They're sitting in the courtroom and they're typing what they hear. The messenger RNA kind of does the same thing. They're going to code the DNA. They're going to take the DNA and they're going to create a new molecule, and that new molecule is going to go to the ribosomes to be translated into a protein. So really with transcription, all we're doing is recopying that code in a different way with the messenger RNA. And here's just another image of that process occurring. Both sides of the DNA molecule, the messenger RNA is going to come and it's going to attach the complementary RNA bases to create that mRNA strand here. Now we have our DNA message transcribed. We have this mRNA that was used from that DNA strand, or created from that DNA strand. Now that mRNA is going to exit the nucleus of the cell and it's going to be translated. We're going to use that code, and think about like if you're translating something for Spanish class or whatever foreign language you take. You're going to take that code that you're given, or you're going to take that word or that phrase that you're given, and you're going to try to make sense of it, and you're going to try to make it usable. That's what we're doing here in translation. We're taking that mRNA code that we got from the DNA molecule, and we're going to translate it into proteins, and those proteins are going to make up our traits. Now with translation, what we see here, we're on a ribosome now. So in your mind, go to a ribosome in a cell. The mRNA is going to come through the ribosome, and there's 3 letters, 3 bases, 3 nucleotides, however you want to put it, from the mRNA, and they're going to make up something called a codon. So a codon is 3 nucleotides. Those 3 nucleotides are going to cause this tRNA molecule to attach to that mRNA, and then it also has an amino acid attached to it. This is AUG. This is a start codon. The transfer RNA is going to bring the complementary base pairs, and those are going to be attached to each tRNA molecule. Each tRNA molecule is going to have an amino acid attached, like I said. Then this is going to keep going through. This mRNA strand is going to keep going through the ribosome, and each time it does, a new tRNA molecule is going to come and attach, and that's going to determine the chain of amino acids. That's how translation works. Remember, we got this RNA from the DNA complementary strand. We transcribed the RNA from the DNA. That messenger RNA is then going to go out of the nucleus. It's going to go onto a ribosome. That ribosome is going to be composed of that, remember, rRNA, ribosomal RNA, and then the transfer RNA is going to have 3 base pairs attached to it, and those 3 base pairs are going to attach to the mRNA, and then the amino acids that are on the tRNA molecule are then going to start to be linked together. And you might be asking yourself now, how does that process work? How does the tRNA know which bases to attach to? Or how do these amino acids get linked together? And you can determine which tRNA molecules are going to bring which amino acids through a codon chart that we're going to look at next. So here is a universal genetic code chart. What you're going to look for here is the messenger RNA codons. Then this is going to give you the amino acids for which they code. Whenever you're looking at one of these charts, I want you to think mRNA. You're looking at what the mRNA is telling you, and that's going to determine which tRNA molecule you're going to get, and that's going to determine the amino acid. I know that might be a little bit confusing, but hopefully as we go through and watch a couple videos and a couple tutorials and do a little bit of practice ourselves, you'll be able to determine this a little bit more easily. If we go back and look at this screen, this first codon, remember 3, this first codon says AUG. That's what the mRNA is telling us. So let's go and look at this genetic code chart. We have A, the first base is A, the second base, can you see that on here? First base, second base, and third base. So we have A for the first base. The second base was U. So we know we're going to be in this block. And the third base was G, AUG. That's methionine or a start codon. The start codon is going to tell the ribosome that this is where we're going to start translating this amino acid sequence. That's methionine or the start codon. That's going to be at the beginning of every gene that needs to be transcribed and translated into a protein. Then we can go back and look at the next example. So just look at this UUC. UUC. Now let's go back to our genetic code chart. First base is U, second base is U, third base is C. UUC, Phe, sorry, I think it cut me off there. UUC, phenylalanine. Let's see if that's what we have here. UUC, the complement will be AAG. That's going to be the tRNA molecule that's going to carry phenylalanine, so we were correct. And you would continue doing that down the whole mRNA sequence, and the mRNA sequence is going to determine which tRNA molecules are going to bind, and the tRNA are carrying the amino acids. When the amino acids are going to link together, that's going to create a protein. The sequence is going to end. That protein will, or that mRNA is going to signal for the ribosome to stop creating that protein when it encounters a stop codon. There's only one start codon, that AUG. Every mRNA strand, every protein sequence should start with methionine or the AUG. And the stop codons are UAA, UAG, and UGA. That's going to signal for the ribosome to stop translating this protein sequence. And this is all known as the central dogma of biology. Everything in molecular biology is based on this process. It's super, super important to know and understand. We have our DNA strand. It is going to replicate to pass on its own genes to its daughter cells or the next generation of cells. And then we have transcription, where we're taking that DNA and we're turning it into RNA. And then for translation, we're going to take that RNA that we got from our DNA molecule and we're going to translate it into a protein. That protein is going to determine your traits. Proteins are made of chains of amino acids, and the way those amino acids are linked together is going to determine the function of that protein. And think of proteins like building blocks. They make up every part of you. They make up all that you are. Some are more functional, like lactase is going to break down milk, and some are structural, like the proteins that make up your muscle or your hair or your skin. Depends on the protein and its function in your body. And a gene is a section of DNA. It's going to have lots of bases, some more than others, and it's going to code for a protein, and those code and those proteins are going to result in a trait. Hopefully that makes sense, and let me know if you have any